In this part of the video, we discuss the process of translation. Translation is a process in which a mRNA is translated into a protein or a protein is made out of an RNA. So translation involves certain machineries. The most basic machinery in the process of translation in eukaryotes is the ribosomes, which consist of two counterparts, that is 60S and 40S. These two are our ribosomal counterpart. Now, apart from the ribosomes, we have a tRNA or transfer RNA that functions to carry the amino acid with it. Now, the functions of the 40S and 60S ribosomes. The 40S ribosomes locates the portion where the start codon or the initiator codon AUG is located. So this is our mRNA. Now, the smaller ribosome locates where the start codon is located. Now, start codon is specific in eukaryotes. That is, it is AUG, which codes for methionine. In this is our start codon. Now, stop codons are of three types in eukaryotes: UAG, UGA, and UAA. Now, why are they our stop codons? It is our stop codon because it doesn't code for any amino acid. That is, once the entire ribosomal apparatus reaches the stop codon, there is a particular factor called the guanosine releasing factor that binds to UAG, and this causes the entire apparatus to fall off. Now, so we get our ribosomes back, our mRNA back, our tRNA back, as well as in the end of the process we get a strand of amino acids that we call as a protein. So what happens is that once the 40S ribosome is bound to AUG, a tRNA carrying methionine, right? Now 60S ribosome consists of three sites: A site, P site, and E site. A site is the active site, P site is the peptide site, E site is the exit site. Now the first tRNA will come and bind to the peptide site or the P site. Now after AUG we have UGC. So our amino acid complementary to UGC is 309. So 309 will come and bind to the A site. Now once 309 comes and bind to the A site, there is a formation of peptide bond between methionine and 309. Now once the peptide bond is formed, the entire apparatus consists of this two ribosomal setup, moves 3 nucleotide forward. So now we search for an amino acid complementary to GUU. Now the amino acid complementary to GUU is glutamic acid. Now, once this apparatus moves 3 nucleotide forward, the tRNA at the P side moves to the E side and the one at the A side moves to the P side to keep the A side vacant for this glutamic acid to come and bind to this. Now, once this tRNA comes and bind to this, the entire setup moves again 3 nucleotide forward, so that the one at the P side moves to E side and one at the A side moves to P side, and a peptide bond is formed between 309 and glutamic acid. So this process continues until you reach one of the stop codons. Here it is UAG. Now once you reach all of the stop codons, our entire setup falls off and we get a strand of amino acid, that is methionine, 309, glutamic acid, and glycine. This is the amino acid we produce from this strand of mRNA. So in this chem 2 we want to represent the same process. So in the next part of the video we are going to make you understand of what the game is about.
So this is a two-player strategy game with each player is given a mRNA sequence, as you can see on the right. Each player has 8 pieces denoting the tRNAs that we mentioned in the biology section before. They are arranged in the back rank. The back rank is this section for each player where the pieces are initially kept. The objective of the game is to move to the codon squares as written in the mRNA sequence. For example, white has to go to AUG, then AUC, then AGC, and finally UGA, which is the stop codon. Translation has to end with the stop codon and it has to start with the AUG start codon. Now we describe the general movement rules. suppose that we have a white piece here and a black piece here. White can move forward, left, or right. Similarly, black can move forward, left, or right. None of them can move backwards towards their own rank. Then there is something called a capture move. For example, white can capture black in this scenario diagonally, provided the square behind it is empty. In that case, the black piece is removed from the board. It is converted into a white piece, and the white player can put it anywhere into the back rank. The capturing piece is moved to the square diagonally behind the captured piece, like this. So, as we discussed before, the objective of the game is to reach the codon squares in sequence and remove them from the board. So once white has reached AUG, which is the start codon, it is instantly removed from the board, signifying the translation has been completed for the AUG codon. It next has to go to AUC, and it's removed from the board, signifying the translation has been completed for AUC. And then the other two codons, AGC, and finally the stop codon, UGA, signifying that the mRNA sequence has been translated. The same is the objective for black. Of course, you can reach the codon squares in sequence and remove them from sequence one by one, as we just described, or you can go to the other codon sequences without going to the start codon, as in this case, in which case the pieces cannot be removed from the board and they behave like normal checker pieces, which can be moved front, left, or right. They can also capture other pieces, and they can also be captured by the opponent's pieces. Basically, they're just normal checker pieces, unless all codons before them has been translated. So as we were saying, the white can choose to go to AUC first, AGC, and the stop codon UGA, but in the end they somehow managed to reach the start codon. In that case, translation begins the moment white reaches the start codon and it's removed from the board. AUC is next in sequence and it's already on site, so it's removed from the board. AGC is next in sequence, it's on site, so it's removed from the board, and UJA is already on site, so it's removed from the board. White wins in one move, provided they can figure out the strategy to occupy all the codon squares before reaching AUG. There's another way to win the game. As we already mentioned, the main objective of the game is to complete translating the mRNA sequence. Whoever does it first wins. Otherwise, you can capture all of your opponent's pieces and convert them into your own army, thereby causing a win by default. That's all.